Welcome to the second video of Unit 2 Sensation Perception. This video is going to be looking at vision. Now, we're really going to be focusing specifically on the sensation side of vision, so we'll talk about the different parts of the eye and then how we actually collect that information, that sensation. Our next video is going to focus entirely on the perceptual side of vision, so we'll start kind of playing with it a little bit and start pulling apart, what do we see versus what do we perceive, and how does all those things kind of work. Now, obviously, we're going to be spending kind of a little bit more time on vision than any of our other senses. Our last video is literally just all the other senses all at once, but we'll kind of get into why that is a little bit as we go here. So the first thing I want to do is kind of start us with a little bit of a test or a little bit of a question to think about. So what I want you to do right now is look at this image on the right side here, and I want you to take a few moments. You can actually pause me if you want for just a second here, and try and find the predator in this image, right? There's a predator in here somewhere, and you need to find it. Okay, so go ahead and pause me right now. Okay, so hopefully you unpaused after a few moments. Now, there's something that's really important to think about here, is the reality is if you didn't find this predator within like a few seconds, you would have been eaten. Okay? Now, let's change our image a little bit here. So I'm just going to move myself out of the way in just a second. Oops, I'm looking on the wrong screen. Move myself out of the way here. Okay, now, if you look at these two images, right, the image are exactly the same, except for one side has color. Now, on the left, the image is actually like a perfect representation of the quality of light, right? How much light energy is coming into your eyes. But it's the exact same as the right. All they have done on the right side is just added in color to that image. Now, on the right, it's obviously very apparent where that predator is, right? It's right here in the bottom right corner. Now, one of the reasons for doing this is, again, thinking about, that idea that we kind of talked about at the very end of the last video, which was we don't see things as they are. We see things as it is useful for us to see. And an example of that we could talk about is something like color, right? Why don't we just see the amount of light, the quality of light? Why do we see color, right? Why is that added in? And one example we can maybe give is something like this, where, obviously seeing predators is very important as a species evolves. So let's kind of jump into vision a little bit more. And here's kind of another image we could actually look at again. So let me go down here to the bottom. So when you look at this image, this is actually a black and white photo, okay? All that is there is lines, and you can see them kind of coming through here, right? You can see them going kind of hexagonally. All they've done is drawn color lines through this black and white image. And what's happening actually is our brain is actually filling in the color into these different spots. Right, so again, this is an entirely black and white image, and then there is just color lines draw through it, and our brain is actually adding in the color of like their shirts, of their skins, of the background, right? All of that is added in by our brain. So again, right, we're kind of trying to pull apart a little bit these ideas of, okay, what do we really see? What are we perceiving? And again, right, it's really important to think about this idea of that sensation versus perception, right? What is actually happening in our eyes? What are we actually collecting? So let's talk about seeing in general. What is it? What are we actually doing when we see? So there's a few things that are really important. The first one is also explaining why this section of this unit is so much longer than the other senses, is that vision is our dominant sense. Vision is the main thing that we use for pretty much everything, right? We have other senses to do other things, right? Like hearing is obviously pretty important, but vision is definitely our most important thing, right? It's really important for us to see things and look at things. We actually have, that's kind of why we have a pretty big chunk of our brain devoted to vision, right? So it's our dominant sense here. So how do we see, right? Basically what we're doing is we're using our eyes to, again, go through that process of transduction, right? Turning energy into something our brain can understand, to turn electromagnetic energy into neural impulses, right? Again, that's our transduction, turning something into something else. So our eyes, all they do is collect electromagnetic energy. Now, there's a whole bunch of different things that fall into what can be called electromagnetic energy, right? And we actually see this chart right here. We only actually collect a very, very, very small portion of what's really there, right? And you've probably seen this before somewhere, of all the different kinds of like rays and different types of radiation that we have, right? So we have like gamma rays, which are incredibly small. We have X-rays. We have ultraviolet rays, which is really common. It's like something that comes from the sun a lot. We talk about ultraviolet rays as well as gamma rays. We have visual light, which is that small little sliver right there. You can kind of see the little rainbow there, which is what we see. We have infrared rays, which is heat. We have radar. We have broadcast bands. We have AC circuits, right? So we've got this huge kind of spectrum of light. Okay, a huge spectrum of, I shouldn't really even say light necessarily, this huge spectrum of electromagnetic energy. The small portion that we see is what's called visual light, right? Which is what we experience. Now notice it's very tiny, right? Now technically, we could have evolved eyes that could sense more things, right? Like, would it be useful to see through infrared rays? Yeah, maybe, right? But we didn't evolve that way. So we only see a small spectrum of what's actually there. And again, that's an important piece of understanding that we experience things not necessarily what they are in reality, but we experience things in a way that are useful for us, right? That are useful evolutionarily. So we have this visual light, which is a small portion of this whole spectrum that our eyes are collecting. So that's what they're doing. They're collecting this light and they're turning it into a neural impulse. So what is light energy? Basically, light energy we can break down into two kind of measurements that are really important for us. Now, again, all of this is waves, right? All of these things are traveling in waves. So there's two parts that are important as far as vision goes for us: wavelength and amplitude, right? Wavelength and amplitude. I'm going to move myself actually some out of the way of that. So wavelength is the distance between two peaks of a wave. Now, we can talk about this for light. We can also talk about this for sound, right? Sound also travels in waves, so we're going to measure it the same way, but it's going to be obviously a little bit different. So for light, right, it's the distance between those two peaks. You could technically measure the distance between the two depressions, I guess you could call them, as well, but you almost always will use peaks, right? So the distance from here to here, okay? Now, wavelength is really important because wavelength determines the hue or the color that we experience, right? So different wavelengths, this distance between those waves are going to determine, right, what color we see. Now, really, the term that we're going to use in psychology isn't color; it's going to be hue. It's going to be hue that we see, and really, it's going to, if you've seen like a spectrum of visual light, right? We can go from violet to reds. Violets and reds. Those are the opposite ends here, okay? And there's actually a really interesting thing. So like this is kind of a little bit disconnected, but you may have heard it if you took like an astronomy class or other physics classes. But this is actually something that we've learned and used in other ways, right? So one thing that we know about like space, for example, is that we know that it's expanding, that everything is going away from itself, right? Or away from each other, basically. And the reason we know that is because when we look at different things in space, right, we can look for these hues in those objects, in those lights, right? And what we know is that if something is traveling towards us, right, it'll have a violet hue. Because if it's coming towards us, those wavelengths are going to get shorter, right? It's going to get mashed up on top of each other, basically. Where if it's going away from us, right, they're going to stretch those wavelengths and it'll have a red hue, right? So we can actually see this in other things as well. But again, if it's farther apart, the farther apart it is, we're going to see more red. The closer together it is, it's going to be more violet. Okay, so again, that's the distance here and here. We also have what's called amplitude. So amplitude is how tall or short, right, that wave is. Now, amplitude is the measurement of the energy of that light. Okay, so the taller it is, and you can kind of see this one that's waving back and forth, right? So when it gets really tall, that's a really high amplitude. That's a high amount of energy in that light. The lower the amplitude, right? So like right when it gets to the middle here and it's really short, or like this one that's really short, the lower the amplitude, the lower the amount of energy there. And what that's going to determine is the intensity or the brightness of the color that we see, right? So the more light, right, the more powerful the light, the more intense and bright that color is going to be. The smaller the amplitude, right, the less powerful the energy, the duller that color is going to be. All right, so basically we can look at a wave and we can say, okay, if let's say it's a really tall but very compact wave, right, it's going to be a very intense violet or blue or something similar to that, right? If it's a very short, very spread out wave, right, it's going to be a dull, right, because it's short, it's going to be dull, not have a lot of intensity, red. Right, and differences in those waves are going to determine what we see. Now, it's really important to understand that, again, this is something that our brain is doing. We're just collecting this wavelength, and our brain is the one that's assigned it the different hues, the different intensities, right? So it's very possible that, like, and we see this actually in other animals, like, not all animals see in color, for example, right? They still probably might pick up wavelength and amplitude, but it doesn't necessarily make color vision for them, right? So all these things are kind of variable. And again, coming back to that idea of we experience the world as it's useful, not necessarily as it necessarily is. So let's start breaking down the eye a little bit and talking about these different parts. The eye is definitely going to be the focus, and vision will be the focus of the sensation questions that you'll see. You will see other stuff, but they're really going to focus mostly on the eye. Again, it's our dominant sense, it's our most important sense, so that's where they're going to do their focus. So here's a nice little diagram of all of the parts of the eye. We're going to go through all of them individually, right? But just to kind of point them all out, we have our iris, we have our pupil, we've got our lens right behind it, our cornea coming around this curved surface right here. We have a blind spot that we'll talk about a little bit later. Our optic nerve comes out of the back. It's not directly out, it's kind of like slightly to the bottom, kind of a little bit of an angle. We have our retina, which is this whole back piece here. And then we have our fovea, which is the center focal point of the back of the eye. Okay, so these are all of the parts of our brain, or I'm sorry, of our eye. And you will be labeling these. There is a coloring book. We'll get there as we finish these parts. So you can use this to help yourself with that as we get there. So let's start breaking these pieces down. What do they do? So our first one is our cornea, right? And this is kind of like a cut out slip of our cornea right here. If you could cut that out, it's this little curved part right here in the front. So notice our eye isn't a perfect circle. It's pretty circular, but this front, there's like a little bulb that kind of sticks out. That little bulb part is our cornea, okay? So what our cornea is actually just a transparent tissue in the front of the eye. And what it does is as rays of light enter the eye, it bends them towards the center of the eye because of this curve, right? So because it's curved, it's kind of like you can kind of think of it almost like a contact lens, although it's not necessarily magnifying, right? As light comes into it, because light travels straight, it bends that light inwards. Now that's really important because as you can kind of see, right, there's only one part of our eye where it's going to actually collect light. We'll get there in just a moment. So it's bending that light into that central area. Okay, otherwise we would lose a lot of the information. It would just kind of like miss. So we'd only get like a really specific tiny little area. So it's actually bending that energy in. So again, really important. It's also a little bit of a protective, right? It makes sure that nothing else gets into these other parts of the eye. But it's this protective layer or this tissue layer. Its main job is to bend that light in. Next, we have our iris and our pupil. So our iris is the colored part of the eye right here. Right, you can see it. In here, right, you can kind of see it. It's right there and right there. They're going to draw it kind of like the tops and the bottoms. Here, there's kind of like a circle. A lot of times, it'll just be like a little liney part that's kind of like indented, okay? But it is a circle, obviously. So what your iris actually is a ring of muscle tissue. And again, this is the colored part of the eye. So if you have blue eyes, right, your iris is that blue color. And it's basically muscular tissue, and its job is to control the size of the pupil, right? And you can see that in this image right down here in the corner. Oh, I forgot I was covering that diagram, sorry. So you can see that this, as our iris, right, basically contracts and expands, or contracts and relaxes, right? You can see that the size of our pupil changes. It gets bigger and smaller. Now what it's doing is the pupil is literally just an opening in the center of our eye that light is going to enter through. So there's actually like not, like the pupil is nothing, kind of. It's just a hole basically. And so again, what's happening is that light's coming in, it's being curved by that cornea and aimed into this opening that's just collecting light. Right, collecting those ultraviolet or those light, that light energy, that electromagnetic energy. Our iris then again is causing that to contract or expand. Right, now what that's doing is it's controlling the amount of light that's going to go in the eye or not. So when it's really bright, we're going to see that iris contract and our pupil is going to get small. Okay, what it's doing is it's letting in less energy. When it's dark, it's going to expand, and it's going to allow more energy in. All right, that's why we're able to, see in the light or the dark, and that's why, like, if you close your eyes and just sit there with your eyes closed for a while, or you have somebody else do this, obviously, so you can see it, and then you open your eyes, you'll actually see that pupil expand, or it'll be expanding, and you'll see it contract, right? Because there's more energy, so it'll darken. Again, that's going to be really important for seeing at night, and we'll kind of talk more about that a little bit later. Then we get to our lens and our retina. So our lens is actually acting just like a lens. If you think about like any kind of lens for anything, a camera, glasses, contacts, even something like microscopes, telescopes, right? All of them use a lens, right? And really what it is it is shaping and targeting this energy. So again, you can see this light energy coming in. It's being curved by our cornea, right? It's going through into our pupil, which would be right here. You can see this little hole. Here's our iris muscles. Here's our pupil. And then it's going to our lens. Here's our lens right here. And what our lens does is, just like a lens on anything else, is it's focusing that energy, right? It's focusing that energy. So if you think of like the lens on a camera, right, where you twist it and zoom in and out, right? It's doing the same thing. It's focusing that energy in. And we'll talk about how it actually, so it actually changes shape. We'll talk about that in a little bit. So it actually changes, like it gets mushed and kind of gets thicker or thinner, right? It does change shape a little bit to help us focus that energy on the back of the eye. Now, what the back of that eye is what's called the retina. The retina is super, 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 super important. The retina is the light-sensitive inner surface of the eye, so it's actually on the inside. It's this kind of like light, I feel like it's a tan color. It's right here as well. And that is going to actually contain what are called rods and cones, and these are the part that are actually collecting the information that the visual information, right? They're actually collecting the electromagnetic energy in the back of the eye. So everything else so far that we've talked about is just about focusing that energy, letting it in. The retina is what's actually doing the transduction. Okay, so it's actually taking that information and then turning it into that neural impulse. Then we get to our optic nerve. So our optic nerve, you can see them right here, right? They're pretty straightforward. They are taking the information from the eye, sending it to the parts of the brain, right? And so you can kind of see the pathway here. Now, there is a little bit of a cross. We've talked about this when we talked about left brain, right brain, and brain hemisphere organization, right? Again, the important thing to remember is that it split the visual field. So you can kind of see that here where this, I feel like this is blue or purple, right, would be the left side of the visual field, or I'm sorry, the right side of the visual field. It's coming in and it's going to that left side of our brain. Same thing here. So you see some information coming from each eye. This will be the left side of the visual field is coming in and it's going over here. Okay. There's a whole bunch of different places this is going to go. Most of it is going to be in the occipital lobe, right? That's mostly where that processing is going to go. But we're also going to have other parts of our brain, you know, obviously doing different things, right? If we're reading words, it's going to have to be our frontal lobe. If they're connecting faces, right, we have our right hemisphere. So we're going to see a lot of different kind of things going on there, okay? But again, all that optic nerve does is it brings that information in and it connects it to the different parts of our brain. Our optic nerve is also really important when we talk about our blind spot. So blind spots, I'm sure you've at least heard of it. You may or may not have really like done much with it. But really all it is it's basically the point where the optic nerve actually connects to the eye. And really what happens here is because the optic nerve is connecting here, there can't be any receptor cells. So there's still light hitting here, but there's just no cells to pick up the information. So what it does is it leaves a little blind spot that we can't see, right? Now we're going to test this blind spot. We're going to do this in class. We'll follow this link. And we'll kind of do a little bit of an activity where you guys get to look at your blind spot a little bit. And we'll also look at how, or kind of explaining why, like right now when you look at something, why don't you see your blind spot? Why don't you see it all the time, right? Like why isn't there just a gap, a little like black spot in your vision? Okay, so we'll talk about that as well, but we'll do this during class. Here's a really good part to like stop and go through and label all of those parts of our eye and also do those coloring pages that are at the beginning of the unit. Okay, so go ahead and pause the video here if you want to do that now. Okay, so now that we have this kind of basic idea of what all these parts are and what they generally do, we're going to talk a little bit more about how do we actually see, right? What is actually going on that's allowing us to see things. So the first thing we want to talk about is visual accommodation, right? And what this is basically how we focus, right? How are we able to look at something really close up and then look at something really far away and be able to see it clearly both times, right? That's really important. If you think about like a camera, and we can see that GIF in the bottom there. Think about a camera, right? You have to change that focus, otherwise it gets really blurred. Now our eyes do that automatically, so the question is how? How does that happen? And the way it's going to actually do that is through changing the thickness or the shape of the lens. All right, and the change of that thickness or the change of that shape is going to then allow that energy to be focused on the back of the eye, depending on if they're far away or close up. Now there's two different ways you can see it, right here, and then also seeing it here. So when we have something that's far away, right, where our lens is basically going to relax, right? You can see it here. It's relaxed. Our lens is going to relax. It's going to basically become elongated. Okay? It's going to become elongated. And what we're seeing here is literally where basically because something's far away, as that energy comes in, this elongated lens is going to then focus it on the back of the eye. We see the same thing with our near point, right? So when we have something that's close, we're going to see that lens kind of get squished, right? And really, actually, maybe this one's just a relaxed eye. This isn't necessarily this. This would actually be stretched further. This is kind of normal, like right in the middle. But here's our close focus, right? So it's basically like squishing that lens. That lens gets kind of squished in. What that does is it makes it thicker, which is going to bend that light more. So you can see it's a little bit more like sharp of a bend in. And again, it's going to focus it perfectly on the back of the eye. So the idea is that as something is closer to us or further away from us, our lens is basically either being stretched out if it's far away or being squished in if it's close up or anywhere in the middle. And our eye is just kind of automatically doing that all the time. It's just adjusting that light and refracting it always on the back of the eye accurately. So that's kind of the, explaining how we can see close up versus far away and why our vision isn't blurry all the time. So the next question is, okay, what's actually going on as far as transduction, right? Like how are we actually getting this electromagnetic energy and turning it into seeing, right? Turning it into what we see every day. So here's kind of the process that we're going to look at. And I actually want to look and see what my next slide is so I can give you, okay. So here's basically the process that's going on. And what this is a few different things. So how does transduction actually occur in the eye? What's actually happening is this light is going to pass through. It's going to get to that retina in the back. And inside of that retina, there's a whole bunch of cells, right? The important ones for us are the rods and the cones, although there are other cells. Those rods and cones are activated by electromagnetic energy, and then they basically pass that down through a few other different cells, which turn it into a neural impulse, which then gets sent to the brain. Okay, so here's what's going on. So here's our eye. Here's that electromagnetic energy coming in, right? It's being curved, it's being focused, right? It's being shot to the back. Here's our retina. So right here's what we're looking at, right? This is this big part. We're zoomed in. So all of this right here is basically like transparent, if that makes sense. Like at least electromagnetic energy can go through it, is basically maybe the best way to say it. So it's actually going to go through like this little wall here. It's going to go through the ganglion cells. It's going to go through our bipolar cells, and it's going to reach the back part of our eye. Okay, and what happens here is that as it goes through this very back part is our rods and our cones. You can see them here, right? So here's our rods. Here's our cones. Now, our cones are a little more triangular. That's why they're called rods and cones. We'll zoom in on them a little bit better in a little bit. Our rods are a little bit more like a rod, if that makes sense. So that's how we kind of separate them apart. And the rods and cones are basically activated by that electromagnetic energy. They're picking up different things that we'll talk about in a minute. They then pass that energy onto bipolar cells, which then pass that information onto our ganglion cells. Now again, the way that these guys are talking is through neural impulses. So that they are turning this energy into an impulse. They're turning it, then sending that energy here. They're sending that energy there. And now it's going through our optic nerve. And now it's going to our brain. Okay? So what actually, the transduction is actually happening in these rods and cones. Okay? Everything else that our eye does is about focusing the light. It's about making sure that that information gets sent into the back of the eye correctly. So let's get a better look at these rods and cones specifically. So first we're going to talk about cones. So cones are light detecting cells, which makes sense. They're how we see. But cones and rods look a little different. They both are kind of doing the similar job, but they're doing it a little bit differently. So cones tend to be focused towards the center of the retina, so this exact back part of our eye. And they function very well in light, well-lit conditions, right? So during daylight, they focus better than at nighttime. And there are ones that are going to detect color, okay? So they're focused in the very back of the eye, in the central part of our retina. They work better during bright, well-lit times, and they are also going to be detecting color. Right? Now, you can see, here's our cone, right? This one looks a little more cone-like. Here's our rod. It looks a bit more rod-like. You're not going to have to, label this. This is just a nice little image for you to see. So these different pieces are really important. So being well-lit is important. The fact that they are sensitive to color is very important. They're also very sensitive to very small details, right? They provide a lot of what's called spatial resolution. So when you look at something like right now, if you look around, hopefully if it's light it's during the daytime, right? If you look around and you look at things, you can see really specific small details, right? You can see texture on things. If you turn the lights off and it's dark and you look at that same thing, you probably aren't able to see it very well. Right? And one of the reasons for that is that our cones are what are actually allowing for that really specific detail. And because they don't focus well when it's dark, basically, even though that information is technically still there, we're not able to pick it up, right? We're not able to see it. In addition, when it's dark, right, our cones, which are detecting detecting color, aren't able to pick up color, right? They're not working very well. So as it gets darker and darker, you see less and less color. even though obviously the object is still the same color, we're not able to pick it up because there's not enough of that energy, right, for our cones to detect it. So we can contrast that then with our rods. Oh, hold on one second. Sorry, I clicked on the wrong spot. We can we can compare that, right, to our rods. So our rods are also, right, photoreceptors. They're also picking up light, this electromagnetic energy, but they work a little bit differently, right? They work much better during low light conditions, right? And here's an actual image of rods and cones, right? That's an actual microscopic images. I do believe these are colored. I don't think they're actually these colors in real life. But rods, cones, they work much better during low light conditions. So obviously they're going to be involved when it's dark out, right? They're also very good at perceiving movement, specifically on our peripheral vision, right? So when you look at like the actual retina of the eye, this is kind of an example of rods and cones, right? Now, there's obviously rods pretty much everywhere in our vision, but outside of the center part, we're not going to have a lot of cones. Okay, we're going to have more and more rods, more and more cones in the middle. So this is kind of an experiment we're going to mess with a little bit, and we can kind of actually pull this apart. But the idea is basically this is why we see the way we see. So this is why when you focus on something, you can see really specific, minute details. You can see color, right? And it's really easy to pick apart. Where if you look straight forward and you go like, if you look at your wall or whatever's in front of you right now, and you focus on one point and you try to think about what's going on like on your periphery, like on the outsides of your vision, right? You're not really able to see super well. Like it's kind of blurry. You don't see a lot of details. The color is probably not super there very well. And this is explained by how our rods work, right? Our rods, which are on the outsides of our eye, aren't really picking up those different things. They're not picking up color. They're not picking up specific details. But they do see movement really well, which we'll see. Additionally, right, when it's dark, all of a sudden our rods are basically going to have to take over, right? And because our cones aren't going to work very well. So when it's dark, all of a sudden we're not going to see details super well, and we're not going to see color very well. Okay, and so that's kind of what we're talking about here. So we're going to pull this apart a little bit. So this is an activity that we're going to do in class, or at least I'm going to explain to you in class, and you guys will do it on your own, where we kind of mess with color vision and we mess with these different ideas of rods and cones as well. So let's talk about how we actually process color. Okay, how is this actually pulled apart here? So there's two basic theories to how color works, right? Now the reason we have this is because there's only so many different kinds of rods. There aren't rods for every color. So the question kind of becomes, how do we see all the different colors we see? So there's two theories, right? One is called the Young-Helmholtz trichromatic or three-color theory. Young-Helmholtz trichromatic theory or the three-color theory. You will need to know the names of these theories, by the way. This is a little tough part to remember, but you do need to know these. So the three-color theory, the trichromatic theory, is that all colors in the spectrum can be produced by combining red, green, and blue. Okay, red, green, and blue are the three types of cones that we have. So the three types of cones are each receptive to one of the colors. Now this is important because we can actually obviously look at, like we can open up people's eyes and look at the different cones, and we know that there's only those three kinds of cones. We know that our cones are only receiving red, green, blue. So the question becomes, okay, if we're only getting red, green, and blue, how are we able to see all these different colors, right? How are we able to see yellow and like black and purple and all these different kind of colors that we have, right? So the idea here is you can kind of see it here, like here's these different cones, right? And they all are interacting with different wavelengths, right? Remember we talked about how wavelength determines color. So each cone is receiving different wavelengths, and that wavelength then is being turned into a color. And the idea here of the Young-Helmholtz trichromatic theory is that basically this information is combined, and then it creates all the colors that we see. Okay, so it's combining this information, and we're seeing our whole spectrum of color vision. Now, there's also what is called the opponent process theory. So again, these are things that we're not necessarily totally positive exactly how it's going to work. Our opponent process theory kind of looks at the young Helmholtz and goes, yes, the fact that there are these 3 different kinds of cones, but it doesn't really make sense. And one of the big questions is like, and any of you who are art people might notice that like red, green, and blue seems off, right? That's not like the primary colors which we use to make everything else, right? It's missing yellow, for example, right? So if there's no yellow, well, how are we able to make these other colors? So the opponent process theory kind of looks at that and tries to fix it a little bit. And basically what they say is there's opposing retinal processes that enable color. So there still are the red, green, and blue cones. These are the cones. But each of them is kind of actually working not just with themselves, but there's also this opponent process that are what are called ganglion cells. So these ganglion cells are also the opposite of those colors, right? So it's red and green, yellow and blue, white and black, right? So white versus black, red versus green, blue versus yellow. Okay? And what's happening here is that some of the cells are being excited by opponent colors and inhibited by other colors. So basically what's happening is there's different cells here that are getting excited, whether it's white or black, green or red, blue or yellow, right? Based on the information coming from those cones. So as that information comes in, it goes to these ganglion cells. Some are excited, some are inhibited, right? That information will then be combined, which will then give us the colors. Okay, so very complicated. The biggest difference is this, and here's kind of how you can maybe remember it. Trichromatic, right? Tri is 3, chromatic you can think of as color. So 3 color is literally just the 3 cones. The 3 cones combine to create all the colors we see. That's pretty much the simple explanation of it. Opponent process, right, has these opponent ganglion cells, these extra cells that are going to be inhibited or excited, right? We've talked about that with neurons before, inhibited or excited, and that's going to determine the opposites. Now again, opposite colors, red, green, yellow, blue, white, black. So you can think about those here. And these are the 2 theories of basically how we actually get color. Because again, all we're receiving is wavelengths of light. That's it. So how do we turn that then into the colors that we see? So one of the things that actually helps us see this a little bit is what are called after images. So after images actually are really useful in supporting the opponent process theory. Okay, so basically what an after image is the continuation of a visual sensation after the removal of that stimulus. Okay, so we're going to try one, and I think maybe doing this example is maybe the best way to explain what we're talking about here and why it supports the opponent process theory. So can I go? Okay, so hold on, I need to shrink myself so I'm out of the way here. I'm going to be really tiny in the corner. Okay, so what I need you to do is there's this white dot right here in the middle. And all I want you to do is just stare at that dot, okay? You can blink, obviously, you don't have to like keep your eyes open, but I want you just to continually stare at the dot, stare at the dot, stare at the dot, okay? As you're staring at the dot, I'm just gonna keep kind of talking a little bit, and in a little bit, I'm gonna switch the slide, okay? So now the important things here are do not look away from the dot, okay? Just continue to stare at it. Now the idea behind after images and why they support the opponent process theory is this. Is as we're looking at this dot, right, you are collecting all of this visual information from what you're looking at, right? So you're collecting that green, you're collecting that black, you're collecting that yellow. And what's happening is those cells are being used, right? So we're seeing, right, those cells are being activated over and over and over again, right? As you keep looking, you're being activated more and more and more and more. Now, the idea is that what happens is when I change my slide, you'll see the same exact flag, but suddenly those colors are flipped, right? Now the greens are reds, the blacks are whites, and the yellows are blues. Okay? So what you're looking at right now, or what you were just looking at, is an after image. So I'm going to go back a second. So that's what the after image is, right? Is this continual visual sensation. So you're still seeing something, even though this slide is completely white, as you can probably see now, right? There's nothing on it. It's just a blank slide. So how this supports a pronet process is that, again, the idea is that kind of like these colors that are being seen in the actual image here are kind of getting worn out. Those receptors are being worn out a little bit. They're being used. So when we then get no image, which, to be very clear, white is not the absence of color. White is all colors. Okay? So when you see this white, what's happening is the colors that we were just looking at, the green, the black, the yellow, all of a sudden, those cells and receptors that should be picking them up aren't. So what we're seeing in this white, which again is all colors, right, are the colors that we aren't basically tired out. So we're still picking up the blue, we're still picking up the red, we're still picking up the white, because those basically were not being used before. Okay, so again, the idea of that is supporting this opponent process theory. So let's go to kind of a few last pieces of our vision here. Let me make myself bigger again. So kind of talking a little bit about how we get this sensation, turn it into perception. Okay. So there's a few different things that are going on. Now again, this is kind of stepping into the perception side of things. So we've collected all this energy, right? It's sent to our visual cortex, and our brain has to then do something with it. So basically what's happening is in that visual cortex, there's a ton of different specialized neurons. These are called feature detectors, right? Feature detectors right here. So these are specialized neurons that are reacting to different things. So it can be reacting to the strength of a visual stimuli, responding to shapes, angles, edges, lines, and movement. So it's picking up all of these different things, and each one is kind of working on its own part. So what's happening then is we've got these feature detectors that are all, doing a different thing for whatever we're seeing. Through parallel processing, which we've talked about before, right, which is our brain doing multiple things at once, all of these parts are going to be basically organized and interpreted at the same exact time. Okay, so for vision, right, we're going to be processing color, motion, shape, and depth all at once, right? And so you can think about it this way, right? If here's our image, right here, this bird, so I'll move that so it's not in the way. Our image of this bird, right? Some of our cells are only looking at the colors in the bird. Well, it would be the colors of everything, but the colors. Some of them are seeing the motion, right? Because we're getting different kind of like, you can think of it like a movie, we're getting a whole bunch of different pictures at once, right? So we can see the change in that bird. Some of them are looking at the form. So where is the end of the bird, the edge of the bird, and then the background behind it. Some of them is looking at depth, right? It's adding, okay, what's closer to me, what's further than me. And all of those parts are happening at once, and then they're combining to be what we see. Okay, which is why, like right now, if you look at your computer, you know that behind your computer is, your wall or your desk or your bed or whatever. Right? And you can see the difference there because your brain is doing all these different pieces at once. So feature detectors are basically, you're not going to have to know different specific ones, but they're these specialized neurons that are looking at these different parts of vision. Okay, we're going to watch this in class. It's basically like a nice little overview. It's actually the Blue Man Group, but it is actually a really good overview of how our vision actually works. Okay, so that's going to wrap up our part 2. Again, go ahead, take a few moments and do a quick summary. If you didn't do it before, it'd be a good idea to go through and label and color in those different images or those different parts of our eye, because again, you have to know all those different parts for sure. Other than that, have a good one, and I will see you guys next time.